Good evening, students. It's almost 11:40 p.m. on 28th November 2023. I took this much time to put up the notes for this lecture to make it easier for you to revise whenever you want. So today we'll be dealing with histology of lymphatics and lymphoid tissue. So in this particular session, we'll be dealing with only two important slides. One will be the lymph node and the other one will be the spleen. So in coming lecture, we'll go ahead with thymus and palatine tonsil. So before going into actual description of lymph node and spleen, we'll be studying some introduction regarding the lymphatic system. You can see here, it's a part of circulatory system. At capillaries, some amount of fluid enters into surrounding tissue as tissue fluid. Some fluid re-enters into circulation at venous end, but remaining fluid is returned to circulation through lymphatics. Lymphatic system includes lymphoid tissue. It's a site of proliferation, differentiation, and maturation of lymphocytes. Example, thymus, spleen. It also includes lymph, fluid passing through the lymphatics, lymph vessels, vessels through which lymph flows. The smallest of these lymph vessels are lymph capillaries, largest will be the lymph ducts, example, thoracic duct. Functions of lymphatic system, main thing is the immunity. The other one is like volume of blood is maintained by retaining of lymph to the circulation. And this lymphatic system also acts as first line defense in air and food passages like Valdes ring. Then filtration of lymph in lymph nodes, then transportation of dietary lipids through lacteals of intestine. Classification of these lymphoid organs based on function, primary and secondary. If you see primary lymphoid organs, production and maturation of lymphocytes takes place in these. Example, thymus for T lymphocytes and bone marrow for B lymphocytes. Secondary lymphoid organs, antigen presentation to lymphocytes and particular group of lymphocytes gets activated. Example, lymph node and spleen. So here also there is production of lymphocytes, but it is not from precursors, it is from pre-existing lymphocytes. Based on structural arrangement, diffuse lymphoid tissue means lymphocytes are diffuse. So they are spread everywhere diffusely. So where we can see those kind of arrangement is in the mucosa of GIT, mucosa of respiratory tract, mucosa of urinary system. So this is only about diffuse. They are not aggregated into some specific thing. So in the same mucosa of GIT, there is aggregation of a lymphoid tissue that is called as mucosa associated lymphoid tissue. Even in respiratory tract, other than this diffuse lymphoid tissue, there is some aggregations of lymphoid tissue called as bronchus associated lymphoid tissue. So this is dense lymphoid tissue, that means free segregated lymphocytes. So whereas the same lymphocytes, if they are densely arranged as nodules, they are these kind of aggregations are seen in specific lymphoid organs, which are called as discrete lymphoid organs: thymus, lymph node, spleen, and tonsil. And in respiratory tract and in gastrointestinal tract, we can see two more aggregations. In respiratory tract, it is bronchus associated lymphoid tissue. In GIT, it is mucosa associated lymphoid tissue. Examples, Peyer's patches. So lymph, it is transudate from blood, contains similar proteins as in plasma but in small quantities. Predominant cells are lymphocytes. Large fat molecules enters lymph vessels from intestine and makes it appearance milky, which is called as chyle. So this transport is done by a specialized lymphatic capillary called as lacteal, which is present in the small intestine. So lymphatic vessels, the smallest of them, lymph capillaries, begin blindly in tissues, structure similar to blood capillaries but with greater permeability. Endothelium, uh, with absent or poorly developed basement membrane, and no pericytes or no connective tissue around capillaries, but there will be some filaments which will be connecting this capillaries to the surrounding tissues. Large molecules enter them through gap between endothelial cells. Lymph capillaries are absent at certain areas like vascular tissues of body, namely cornea, hair, nails, and even splenic pulp and in the bone marrow. You can see here, this is the heart, this is the arterial system. You can see the arterioles, then the capillaries. These are the venules, and this is the vein. You can see here, there is some amount of transudate from the blood, which is being collected by these blind-ended lymph capillaries, which are draining into lymphatic vessels, then lymph trunk and lymph duct, and ultimately it is going back to the veins, then to the heart. So this is actually maintaining the volume of the blood. You can see here, this is an enlarged view. It is the end of a lymph capillary where it is beginning, and you can see some filaments anchoring the lymph capillaries to the surrounding tissue. You can see these are the lymph capillaries surrounding the blood capillaries. So after lymph capillaries comes the lymph vessels proper. Structurally is similar to veins. Tunica intima, media, and adventitia can be differentiated. Elastic fibers are prominent in all three layers. Smooth muscle cells seen in media and adventitia in majority. They are arranged circularly, but in thoracic duct, they are arranged predominantly longitudinally. Valves are present more in number than in veins, gives beaded appearance to the lymphatic vessels, guides the flow in one direction. So, lymph capillaries, then lymph vessels proper, then further larger things like lymph trunks and lymph ducts. So, jugular lymph trunks, subclavian lymph trunks, these are few examples for lymph trunks. Then lymph ducts, right lymphatic duct and thoracic duct. Thoracic duct is the largest lymphatic duct. Arrangement is similar to other lymph vessels except that smooth muscle is arranged predominantly longitudinally. You can see here, this is a cisterna chyli, from apex of which starts the thoracic duct. You can see here, it is ultimately draining into the left venous angle. And you can see this is the right lymphatic duct draining into the right venous angle. So then lymphocytes, just to revise, T lymphocytes, lymphocytes which mature in thymus, four types: helper cells, cytotoxic cells, memory cells, and natural killer cells. B lymphocytes, arising from stem cells of bone marrow. B stands for bursa of fabricus, which is a diverticulum of cloaca of birds where B lymphocytes are formed. B lymphocytes seen in lymphatic nodules, especially in the germinal center, are actively dividing, and in the same lymphatic nodules in the periphery, there are resting B lymphocytes. We'll see when we deal with the lymph node. So functions of lymphocytes: antigen recognition by T lymphocytes, antibody production by B lymphocytes, and mainly the immunity, humoral immunity, which is mainly through antibody mediated, and cell mediated immunity, which is cell mediated. So coming to the lymph node. So these are bean shaped, approximate size 1 into 2.5 centimeters, more than 700 to 800 present in the body. Secondary lymphoid organ, specially designed for lymph filtration. So there is interaction between lymphocytes and bacteria in lymph nodes. Many afferent vessels enter through the convex surface. Only one or two efferent vessels leaves the hilum. Arteries and veins pass through the hilum. You can see here, this is the structure of a lymph node, bean shaped. This is the convex surface from which many afferent vessels are entering. So this is the hilum through which one or two efferent vessels will leave, and the artery and vein will enter. This is the central part, medulla, and this is the peripheral part called cortex. You can see this is the capsule, and these are the trabeculae, and this is the subscapular sinus, and this is the cortical sinus. These are the medullary sinuses. So we'll deal with them further. So you can see the components are briefed here: macrophages, lymphocytes, B lymphocytes, reticular cells, and fibers. So if you see basically lymph node, these are the points you are going to understand. You should know what is the capsule, subcapsular sinus, trabecular sinus, cortical sinus, and what is the cortex, and what is lymphatic nodule or follicle, and out of that what is primary and what is secondary, then the germinal central. So within then what is this mantle zone or corona, paracortex, medullary cords and sinuses. These are the points under which we are going to see the microscopic anatomy of lymph node. You can see here what is the cortex. It is the outer zone of densely packed lymphocytes which stains darkly and will not extend into the hilum. Then medulla is the light staining zone of the fewer lymphocytes in the center. Capsule, which is rich in collagen fibers, elastic fibers, and some smooth muscles may be present. Then the subcapsular sinus beneath the capsule. Trabeculae, these are the septa from the capsule extending into the node and divides the node into lobules. Hilum, occupied by mass of dense fibrous tissue where there is exit of efferent vessels and then entry and exit of artery and vein. So then reticular fibers form the framework to support other components of the lymph node. Then what is the cortex? Cortex is rich in lymphatic nodules or follicles. Primary nodules are aggregation of small lymphocytes. Here there is actually no response of lymphocytes to the antigen. That's why they are primary nodules. Secondary nodules are nodules with germinal center. This is actually happening because it has been exposed to some kind of antigen. So germinal center is the central light staining region of the proliferating lymphocytes, otherwise called lymphoblasts. Germinal center indicates the response of nodules to antigen. Mantle zone or corona is the small lymphocytes encircling the germinal center. I can show you. So this is the capsule with subcapsular sinus. This entire thing is the cortex. This is the medulla, where you can see these are the medullary cords and these are the medullary sinuses. So if you see, this is a lymphatic nodule or follicle where in the center you are seeing the light staining germinal center, and you can see this is the mantle zone or otherwise called as corona. This is the mantle zone, otherwise called as corona, surrounding the germinal center, and this is the peripheral dark area where resting lymphocytes are present. So this is an enlarged view. This is the light staining germinal center where active B lymphocytes are present. So this is the mantle zone, you can see, which is surrounding the germinal center, and this is the peripheral zone where there are resting lymphocytes. And you can see this is the capsule and this is the subcapsular sinus. And here you can see the variability of the cortex. So in the absence of antigen stimulation, see this is a primary follicle. In the presence of antigen stimulation, you can see this is a secondary follicle where you can see there is a germinal center, mantle zone and the peripheral zone. So in cortex we can see the subdivisions, like superficial part of cortex is rich in nodules, so called as nodular cortex. Then the other part is the paracortex, which lies between nodular cortex and the medulla. It is mainly populated by major portion of T lymphocytes of the lymph node. Paracortex is also called as thymus dependent cortex. You can see now, just as an overview, this is the capsule, this is subcapsular sinus, this is the lymphatic nodule with the germinal center, this is the trabeculae, and you can see this is the nodular cortex and this is the paracortex, and you can see this is the medulla, these are the medullary sinuses and these are the medullary cords. So medulla, branching and anastomosing cords of lymphocytes are present, which are called as medullary cords, and these cords are separated by medullary sinuses. So in the medulla, we can see the lymphocytes, macrophages, reticular cells, dendritic cells, and reticular fibers, which form the network for supporting the cells. So you can see here, enlarged view. So these are the medullary cords and these are the medullary sinuses. Circulation through lymph node. So first the afferent lymph vessels will reach the lymph node through the convex surface. Then they pour the lymph into the subcapsular sinus. This travels through the trabecular and cortical sinuses and then enters the large medullary sinuses. One or two afferent lymph vessels will leave the hilum. So lymph passing through sinuses comes in contact with macrophages, which remove bacteria and particulate matter. So lymphocytes really enter or leave through these channels. Arteries enter through hilum, pass through medulla to reach the cortex. So here they form arterioles and capillaries, which are arranged as loops that drain into venules. So post-capillary venules in lymph nodes are lined by cuboidal cells, hence called as high endothelial venules. Usually the capillaries should be lined by a flattened endothelium, whereas here they are lined by cuboidal cells. So they are called as high endothelial venules. So they allow passage of lymphocytes between bloodstream and surrounding tissue. This is actually receptor dependent. You can see the receptors on endothelium and their interaction with lymphocytes allows the passage of lymphocytes. So functions: lymphocyte production, both B and T lymphocyte produced from pre-existing lymphocytes, not from stem cells. It is from pre-existing lymphocytes. Then removal of bacteria and particulate matter, immune response sites to antigen. Then plasma cells, which are the fully mature B lymphocytes, produce antibodies. Then T lymphocytes attack cells that are foreign to the host. So then coming to our next topic, spleen. So if you see where it is located in left epigastric region between 9 to 10 ribs. The longitudinal axis passes through the 10th rib. Largest lymphatic organ in the body, can vary considerably in size and weight. So blood forming organ in fetal life and blood destroying organ in postnatal life, otherwise called graveyard of RBCs. Filters blood, blood-borne antigens and microorganisms. So structure, there is a serous coat which is nothing but the peritoneum covering the spleen except at the hilum. Then below this serous coat there is a capsule which completely encloses the spleen. So trabeculae from the capsule extend into the substance of the spleen and form network. See, there is no subcapsular sinus in spleen, whereas there is a subcapsular sinus with lymph node. So elastic fibers are abundant in capsule and trabeculae. Elastic fibers are abundant in capsule and trabeculae. Space between trabeculae is filled with reticular fibers and amorphous matrix. So this space is also having fibroblasts, macrophages in relation to reticulum, and between the reticulum there are blood vessels, lymphocytes, blood cells and macrophages. You can see here, this is the capsule. See the absence of subcapsular sinus. These are the trabeculae, and you can see this is the white pulp where there is an eccentric arteriole. So these are called as periarteriolar lymphatic sheets, and this is the red pulp where there are splenic cords and splenic sinuses. You can see within the trabeculae, you can see an artery and a vein, trabecular artery and trabecular vein. So this is also called a central artery, but still if you observe in this properly, this is eccentric in position. So in relation to this, we can also see a splenic nodule. Splenic nodule. If you observe difference between lymph node and spleen, here there is no subcapsular sinus, and in this splenic nodules, you can see there is an eccentric arteriole, which is absent in case of lymphatic nodule in lymph node. Circulation through spleen. Splenic artery reaches hilum and divides into 5 branches. So branches travel through trabeculae and subdivides. Arterioles from trabeculae pass into intertrabecular spaces. So for some distance, these arterioles are covered with dense sheet of lymphocytes called as white pulp. You can see here. So these are the arterioles covered by dense sheet of lymphocytes called as white pulp, also called as periarteriolar lymphatic sheets. So arterioles divide into straight vessels called pencillae. So pencillar arterioles can open into red pulp. So that is directly opening into the reticulum where they are directly exposed to macrophages, that is called open circulation. Or the same pencillar arteries can open into splenic sinusoids, which is called as closed circulation. Ultimately, even if this pencillar artery open into red pulp, from this red pulp is also having the splenic sinusoids. So after the blood is exposed to macrophages, then it again enters into splenic sinusoids. So before entering splenic sinusoids, if the blood is in direct contact with the splenic cords where it interacts with macrophages, that is called open circulation. If the blood does not pass into the reticulum but directly passes into splenic sinusoids from capillaries, then it is called closed circulation. So you can see from veins from red pulp or splenic sinusoids enter into trabecular veins or drains into trabecular veins. So splenic sinusoids lined by elongated banana-shaped stave cells. So usually the splenic sinusoids or any sinusoid should be lined by endothelium. Here this endothelium is modified as elongated banana-shaped cells called as stave cells. So what is the main function of this is they can alter their shape to open or close the gaps between adjacent cells because of presence of special fibrils in their cytoplasm. So spleen acts as filter for overnight RBCs. Normal RBC changes their shape and pass through the spleen, whereas aged RBCs cannot alter their shape and get stuck in spleen and later undergo destruction by macrophages. So now splenic parenchyma. So where we should clearly understand the difference between white pulp and red pulp. So white pulp is nothing but aggregation of lymphocytes around small arteries or arterioles. So in H&E stain, it appears basophilic due to dense heterochromatin of lymphocytes. Within white pulp, the branch of the splenic artery or arteriole is called as central artery, though it is eccentric in position. So periarteriolar lymphatic sheaths are the lymphocytes surrounding these arterioles. You can see here, this is the white pulp. This is the white pulp and this is the red pulp. So localized extensions of periarterial lymphatic sheaths is the splenic nodule. So splenic nodule with germinal center is called as Malpighian body. More than one arteriole may be present in relation to one germinal center. So periarterial lymphatic sheaths are rich in T lymphocytes, whereas germinal centers are rich in B lymphocytes. So you can see here. So this is an eccentric artery or arteriole, and you can see this is the germinal center. So this are the white pulp. But if you see the remaining area, this entire thing is having the splenic cords of Bildroth and splenic sinuses. So this is the red pulp. So this is a white pulp and this is a red pulp. So what is red pulp? It is a modified lymphatic tissue infiltrated with all cells of circulating blood. It appears sponge-like, which is lined by reticular cells. So intervals between spaces filled with lymphocytes, macrophages and blood cells. Splenic sinusoids lined by endothelial cells which have processes making contact with adjacent cells. These cells are arranged as cords, splenic cords of Bildroth. So zone of red pulp surrounding white pulp is marginal zone, which is rich in antigen presenting cells. So if you see in this diagram, so this zone of red pulp surrounding the white pulp is rich in antigen presenting cells. Rich in antigen presenting cells. So this is the white pulp, this is the surrounding zone which is called as marginal zone. So this is actually the part of red pulp, but it is surrounding the white pulp. So this is rich in antigen presenting cells. Now we can see clear differentiation here. So this is the central arteriole with periarteriolar lymphatic sheath, and you can see this is an extension from this which is forming a splenic nodule with the germinal center. And here you can see these are the splenic cords of Bildroth and these are the splenic sinuses with blood. So antigen presentation in lymph node is through lymph from tissues. See the point? Antigen presentation in lymph nodes. Lymph nodes are actually receiving only the lymph from tissues. So whereas in spleen, the antigen presentation is directly from blood. So if the patient undergoes splenectomy with any reason, it reduces the ability of that particular individual to deal with bloodborne infections. So central artery, like in white pulp, divides into straight arterioles and capillaries. Some capillaries are lined by macrophages and are called as sheathed capillaries. You can again see here, this is the trabeculae, this is the white pulp, where you are seeing the splenic nodule, and this entire thing is the red pulp. So blood from capillaries opens into reticular meshwork of splenic cards. Then it goes into splenic sinuses. This is open circulation. First, it is opening into the reticular meshwork where the blood is directly exposed to the macrophages. So this necessarily exposes the blood to macrophages before reaching sinuses. So whereas capillaries, when directly open into splenic sinuses, it is called closed circulation. See the difference. Ultimately, capillaries are opening into splenic sinuses, both in open circulation as well as in closed circulation. But in open circulation, before reaching splenic sinuses, the blood is being poured into the reticular meshwork of splenic cards. So sinuses drain into wide sinusoids and then into trabecular veins. So functions: immune response against antigens present in blood, but not against antigens present in tissues, which is seen in lymph nodes. So destruction of damaged or aged RBC. Macrophages also remove worn out leukocytes and bacteria. So in fetal life, production of all blood cells.